Hello everyone. On behalf of the GATS tutoring program, I would like to invite all the students to make use of a new initiative, which is the AP tutoring video series. So what we've done in this is our qualified students have basically broken the AP subjects unit by unit into simple 10-minute videos. We hope that these videos will come in handy for your test prep or for the AP exam prep. The subjects that we have worked on so far are AP World, AP Biology, AP Human, AP Psychology, and AP Calc AB. So if you would like to watch more videos like this, please be sure to subscribe to our GATS YouTube channel. And also, if you would like to be part of this initiative, please email us at gats-tutoring@gatamilsangam.org. Thank you.
Welcome back to Unit 3 of AP Psychology. This is Sensation and Perception, and this part of the exam contains around roughly 6 to 8% of the total AP exam. So the first main thing we need to know is sensation, which is just biological sensory inputs. So what that means is if I touch a piece of wood and I feel that it's wood, that is my brain sensing that that texture is wood. And sensation really has 3 steps, right? First is reception. You gather information from your fingertips, I guess, in this case. Then is transduction. So the stimulus gets converted into neural impulses, so that feeling of texture is converted, and then it's finally transmitted to the brain. So those sensory neurons are sending that information all the way up to my brain. Another thing you need to know about sensation is the 2 types of processings. So there's both bottom-up processing and top-down processing. Bottom-up processing is when I go from the bottom up. So when let's say I'm reading a sentence or reading a word really, and I don't know what it is. As a kid, I'm going to sound out each individual letter. Like if I have cat, then I'm going to sound out C-A-T, cat. That's bottom-up processing. You're basically like, it's like you take the foundation and then you build up from there. Whereas top-down processing is knowing, oh, I've already seen this word before, and it's looking at the whole picture, and then it's coming to the conclusion with that. Another thing you need to know is absolute threshold, which is the minimum stimulus required to be detectable 50% of the time. It's essentially the, if I hear a sound like very, very far away, and I can hear that sound around 50% of the time that it's played, then that means that it's detected, and that means that the stimulus is there. Subliminal stimuli are stimuli which are just in the brain, but they're below the absolute threshold. So they're there, but the brain doesn't want to care about them. So like if it's really, really far away and I can't actually hear it, the brain is still going to detect that stimulus. It's just going to say, oh, it's not necessary. It's just, it's not necessary for my brain to know that. So me to know that. So the brain has that stimulus. It just doesn't send it to you. The signals are just not enough to actually like for you to perceive it. Another important concept is the difference threshold, which is essentially the minimum difference to be noticeable. And this is where Weber's law comes into, which is there's Weber's law on the side right there. You don't actually need to know how to calculate using Weber's law. You just need to know that the idea of Weber's law. And the idea is that, let's say I'm lifting 500 pounds and I add a 5-pound weight on top of that, the chance of me detecting that is very, very low. But let's say I'm lifting 10 pounds and I add 5 pounds on top of that. Well, then the difference is going to be very, very, like, it's going to be easy to recognize. So that's the idea of Weber's law. It's that the more the more stimulus there is, the harder it is to detect a change in stimulus. Another important concept is the signal detection theory, and essentially it says whether we detect a stimulus or not. And that's realistically, it depends on a ton of factors. So whether we detect a stimulus or not is based on what our level of arousal is at the time, what our mental state is like at the time. All those things come into play with signal detection theory. All right. Two other concepts is selective attention and selective inattention. So selective attention is the fact that we can only really focus on one thing at a time. Try to focus on multiple things at a time, you're really going to struggle because that's not a thing that you can do. Yeah, there's no such thing as multitasking. You just do two things worse, is what we like to say. And then the cocktail party effect is essentially if you're in a really loud party and your friend is saying something to you, like they're sitting right next to you, and assuming you can hear them and they're not whispering, then you're able to zone in on one thing, even though there's a lot of different stimuli going around you. That's the cocktail party effect, the ability to zone in on one thing, even if there's multiple, like a ton of other things going around. Selective inattention is the exact opposite. So it's what we miss due to inattention. So say there's a magician and they use a sleight of hand to kind of misdirect you, and then they do whatever magic trick they're doing. That's selective inattention. Your attention is on whatever they're doing with their hands and not on the magic trick itself, and that's why you miss what's happened and what the magic is, and that's why you're confused when it actually the magician completes his magic trick. So that's selective inattention. Another important thing is change blindness, and it's essentially our blindness to whatever changes in a room. If you ever tried to try to complete a spot the difference without the original paper, it's very, very difficult. So that's change blindness. It's our difficulty in figuring out what changes in a situation, really. Okay. Now let's talk about visual sensation, and first is color. So color is seen in varying wavelengths. Essentially what that means is the higher wavelength, the higher on the spectrum it is. So higher, it goes up to violet, and if it's lower, then it's red. So and then everything else is on a spectrum between that. And brightness is the varying wave heights. So all color is waves. I mean, really, the vision is in waves. So brightness is the varying wave heights of that of those light waves. And then lens accommodation is how we actually allow for focusing on things that are nearer and farther. So if I'm looking outside and I see something in the very, very distance and then something that's really close to me, I'm able to focus on those things that are very far away from me. That's lens accommodation. My lens is actually going to stretch to allow me to accommodate for that. The retina is a part of the eye that sits on the very, very back of your eye, and it has two receptor cells. It has rods and cones. Rods are dark and blurry, and they're on like the outside of your retina. So they're really they're on the outside of your vision, basically. And cones have color, and they're on your fovea. The fovea is like your focal point. That's where you see the most color. It's really just the place that's right in front of you, and rods and cones are in the sides of your eyes. You also call this the peripheral vision, because your vision isn't as good there, but it still exists, and you can kind of see, but you can't fully make out stuff. Now, the funny thing is you can't actually perceive color really well in your peripheral vision. So if you've seen an object already, it's pretty easy to make the color, but I want you to try this if you want to. You can take a colored pencil, put it behind your head, and try to see when that color, like that colored pencil actually becomes colorful. Like that you can see it, and make sure you don't know the color of the colored pencil before, because then it doesn't work. And you'll see that you can't actually see that it's colored until you move it in the range of your fovea, really. And the process of visual transduction really goes from rods and cones, bipolar cells, which are and ganglion cells, which convert the receptors into neural impulses, which then send it to the optic nerve, which sends the neural impulses to the supercells in the occipital lobe, which is in the really back of your head. That's just a thing. Yeah, the occipital lobe is the place that recognizes vision and is also conveniently placed in the back of your head. Okay, now let's talk about color vision theories. This is just a fact. If all the light waves are emitted back, then that means that's white, that object's white. And if all the colors are absorbed by a certain object, then that object is black. That's how white and black work. And then everything else is just, so say it's a blue object, then that means it's reflecting blue light and your eyes see that blue light. It's absorbing everything else that reflects that blue light. So the trichromatic theory says that every object is made out of three main colors, right? Red, green, and blue. And you have red, green, and blue cones. And the combination of those cones will create different colors. Whereas that explains a lot of things. Like if you've seen your computer screens are all red, green, and blue, and they make combinations of pixels to display different colors. But that doesn't just explain the reason. If you've ever seen an illusion where you've looked at an object for a really long time, you see the opposite color, that doesn't really explain it. So the opponent processing theory explains this. And essentially, we have really two different rods that perceive two different colors. So let's say I look at a red image for a really long period of time. I'm going to see the opposite image. I'm going to see a green image when I close my eyes because those red cones are very overworked. So the green takes over. That's kind of how it works there. All right, vision constancies. So there's really 5 vision constancies that you need to know. So first is perceptual constancy. So even if an object is changed in a way, we still perceive it the same. Second is brightness. Even if there are different shades, they still look, there's different shades. Even though like they're the same color, we'll see different like shades of light. Third is shape constancy. There's essentially the same shape no matter what angle you put it in. So actually, if you take a piece of paper and you rotate it slightly, it's actually a trapezoid. Like visually in a 2D field, it would be a trapezoid because it's longer here. But your brain still perceives it as a piece of paper, so it's a rectangle. Color constancy is the fact that objects are the same color no matter what the context and surroundings happen to be. And size constancy is when we judge objects based on their size. So essentially, if I have a mouse that's really, really far away from me and I see it really, really big, like it's a really, really big mouse, I'm going to think it's closer than it actually is. Like, let's say it's a big mouse, but my definition of a mouse is that it's small. So I'm going to think that mouse is really close to me because it's even if it's really far away from me. Same thing if I have a really, really small elephant, like let's say it's this big, and I see it really close to me, I'm going to think it's far away because it's an elephant, it's supposed to be really big. So we judge objects based on their relative size. Another important thing is monocular versus binocular cues, and this is essentially how we navigate our world through vision. So there's really 2 binocular cues that you can use. One is convergence and retinal convergence, which essentially when you look with your 2 eyes, there's a spot where those 2 eyes converge, and that's when our eyes will move in the same direction and see the same things. Retinal disparity is when your eyes both see slightly different, which you can try switching from one eye to another and you'll see that slight shift. So you can actually have a better idea of where things are based on those retinal disparities. Your brain will use those to get a better sense of where objects are. So people that have one eye like taken out, it's a little bit more difficult for them to have a perfect sense of where objects are. Interposition, this is a monocular cues, by the way. But interposition is when something is blocking something else. So essentially, let's say I have a car and it's blocking another like very large object. I'm going to think that object is larger than the car because the car is, well, it's blocking it. The car is a big object, so it has to be larger than the car. Another thing is linear perspective. So linear perspective is when converging lines generally tend to converge as the farther they get from us. So if you ever seen rail lines, or if you've ever seen anybody draw rail lines, they converge, like they come to a point when they're really far away from us. So things come to lines come to a point when they're far away from us. Another thing is relative size. So generally far away things are going to appear smaller and close things are going to appear bigger. Shading is, that's just a thing that, I mean, if you've ever seen artists, they can shade stuff. I mean, that happens in real life too, but the shading is just a different colors. I mean, different colors that you can perceive. That's something you can perceive with one eye. Monocular cues, remember, are things you can perceive with one eye. Binocular cues require both eyes. Vertical cues are essentially things that are taller appear taller. That's all it is. We can perceive that. Relative motion is that objects moving at the same speed look the same at different distances. So even if let's say I'm running like 12 miles per hour very far away and I'm running someone else is running 12 miles per hour really close to me, it's still going to look like the same speed. And relative height, the moon generally looks larger on the horizon than up above, because that's just a way of that our brain tricks us, because it's not actually larger, but it looks larger because on the horizon it's it's far away from us, whereas up above it looks to our brain it looks the same. So these are all things you can perceive with just one eye, whereas binocular cues you need two eyes. Okay, now let's talk about auditory sensation. So first is the outer ear, which is essentially the outer area, which is what we see. The middle ear is the more interesting part, and it consists of the hammer, anvil, and stirrup. And these are bones that amplify sound inside the ear. The inner ear is the cochlea, which has essentially hair cells and also liquid that creates vibration. And that gets sent to the auditory nerve, and that gets sent to the brain. And hair cells will generally die if sound is greater than 85 decibels. So this is why when you go to a loud concert, you might feel your hearing is gone, because some of your hair cells will die, and also just because your cochlea and all the inner, the important muscles inside your ear are overworked. Auditory localization is a cue, which essentially lets us know where sounds are coming from, and it depends on volume, timing, and pitch. And our brain can essentially figure out where sounds are coming from depending on those three indicators. And some interesting things about sound waves are that wavelength frequency affects pitch, wave amplitude affects volume, and complexity affects the tuning. So there's different types of tones. So there's pure and complex tones. All right. Lastly, we're going to talk about deafness and hearing theories. So place theory is the theory in which pitch is based on the sound, where the sound hits the basilar membrane. Whereas frequency theory is the idea that hair cells will send signals faster if there's more sound, which leads to higher frequencies. That's what you hear, higher frequencies inside the brain. There's really two types of deafness that you need to talk about. There's conduction deafness and nerve deafness. So conduction deafness is when there's no bone damage, but the actual physical structure inside your ears, like the hammer, anvil, and stirrup, are struggling to produce send these signals, which essentially means you've been to too many loud concerts, and your brain, your whole ear has taken a bit of a beating. So the easy way to fix this is just getting a hearing implant. And nerve deafness is when there is nerve damage, so there's no signals being sent to the brain, and a cochlear implant will generally solve this problem, because the cochlea can send the nerve signals to the through the auditory nerve, and then that goes to the brain, and then it can be processed. Okay. Other senses that we need to talk about. So touch, the skin perceives pressure, temperature, vibration, and pain, so it can do a lot of different things. You've probably felt all of these, so they don't really require too much explanation. Gustation, which is another just fancy word for taste. The only thing you need to know about this is that taste buds can actually grow back. So, and then the other thing is that, there's top-down processing with gustation. So 50% of your taste or something like that is based on how you perceive the food that you're eating. So if you think the food looks good, or its texture feels good, and the smell of the food is good, then the food actually tastes better. Vice versa, if you think the smell is awful, then the food's also going to taste worse. And lastly, smell, which is the only sense that's connected straight to the brain. The olfactory nerve right here carries smell signals all the way up to the olfactory bulb in the brain. And the reason for that is smell is really important in identifying threats. What berries are, back in our hunter-gatherer days, what berries are good to eat, what things are safe and what are not safe. Those things are all based on smell. So smell is linked directly to the brain because it's an important sense. Other senses that we actually do have other senses. We have the vestibular sense and the kinesthetic sense. So kinesthetic sense is the ability to sense our movement, which is something you don't really think about. But if you think about it, when I'm walking, I don't think about where I'm going to place my foot. That happens automatically. Our brain knows that. So that's one of our senses. The kinesthetic sense is our ability to sense where our limbs are without actually like thinking about it consciously. And our vestibular sense a lot is our balance sense. Essentially, that's what if you like rotate, if you've ever been on like those roller coasters where they rotate you really fast and then you feel really dizzy afterward, that's because your vestibular sense was messed up, and that fluid in those ears, the fluid in the ear, in the cochlea was moved really rapidly. So that's what happens. And when this fluid is moved, the body knows where it is relative to the ground because of gravity. So if I'm like upside down, then the fluid is going down. So I know that I am, upside down. That's how the body knows where it is relative to the ground. Okay, lastly, we're going to talk about perception. So there's this really weird phenomenon that you need to know called the phi phenomenon, and it's a series of images that's seen as movement. So if you ever watch stop motion films, that's just images, but it's images there's so many images that it's seen as movement. If you look at this loading picture, that's the phi phenomenon. That it looks like there is a loading symbol right here, but realistically it's just dots flashing on and off. But because the movement is going around in a circle, it looks like those dots are moving. Another important concept to know is the figure ground perception. So there's essentially based on where the foreground is and the background is, we perceive things differently. There's also grouping patterns, right? So this has to go with the Gestalt principles, and there's really 4 grouping patterns that you need to know. One is proximity. So we really, as humans, we want to make things that are close together, we want to group them together. Things that are similar, we want to group them together. Things that are continuous, so if we see a line moving together, then we want to group that together. We want to say that's one continuous line. Let's say there's dots moving together. We want to say that there's continuity there to left. So there's that there. And then lastly, we have 2 more things. We have parallel processing, is when your brain processes multiple things at the exact same time. So this is like when I said you couldn't multitask, you technically can. It's just worse. Parallel processing, your brain is processing 2 things at the same time. So if you're talking and also looking at something, that your brain can do. Now you can't do like 2 complex tasks at the same time. Synesthesia is when one sense triggers the other, and this is something that happens to some people where they have an emotional response to a certain color or a certain a sense. So let's say the color, for example, the example here, the color red allows you to sense roses even though there's no rose. So that's essentially all that we have for perception. And that is also all we have for the Unit 3 sensation and perception. So I'll see you next time in Unit 4 learning.